What? No, they're gonna be fine, Mom. They're gonna be fine. All gonna be okay. You just get out of here. I'm not gonna leave you to it. Now I take it out. I'm gonna start playing my golf. All right. Love you. Love you too. No, no. You didn't want it, and you just got it.
Small measure, she paused as if for emphasis. Make good use of this time out, and I hope one day we will meet again. She inclined her head and then turned back to her dog, who lay obediently on the carpet. Come, Daisy. It's time to go home. Dressed only in a waistcoat and shirt sleeve.
Kyle, would you stay after class for a minute and talk to me? Do you hear and see me? Can you hear and see me?
Yes.
Okay. Any questions or concerns?
No, I don't think so.
Okay. If you go on D2L, if you can, when you are able, look at D2L and make sure that I have your exam grades. I started grading the case studies last night. Over the next couple days, I will finish them. I can't say I'll get them finished today, but I will over the next couple days. At least the first case study. The major thing that I saw is you need to put your answers in your own words. When I'm trying to say this in the kindest way possible, I ran into several papers where the same people had the same answer word for word. And that is not, I don't think that's really physically possible unless you copied them off someone else. And I put it in the comments if I found it, because there's not a lot of people in the class, and when I see an answer that looks exactly identical to one that I just read, it doesn't work very well. It doesn't, if it's right or if it's wrong, it doesn't matter at that point. And one of the answers wasn't really right. And so with these case studies, they are open-ended short answer questions. You need to answer using your own words. And if you aren't, I'm going to dock you points. If I wanted to be really, really mean and awful about it, I could fail you because it's plagiarism to do that. But I'm not going to do that because I don't feel like, I don't know if you don't recognize that what you're doing isn't right, but it isn't appropriate for a college level course to be doing those kind of things at all. And so you can discuss the answers with your peers, but you can't copy their answers, because it's, when it's several sentences or several words. I think the one was, I think it was 7 words, and the same answer was given to me 3 different times by 3 different students. And it wasn't even completely correct. And so I'm asking you, and I'm telling you, do not do it on any homework assignments, because it's not appropriate behavior, and you can be failed in a college class for doing those kind of things. it is considered plagiarism. And it is considered cheating. So please don't do it. I didn't see it on you. If I saw it, I wrote it down on your paper. If you didn't see it, then it didn't happen. And I haven't graded everyone's. I'm probably a little over halfway through with our class, this class. But you can't do that kind of stuff. Yeah, I'll get this one graded. I will work on grading the next ones, and I can't say that I'm going to get it done before the Thanksgiving vacation, but I'm going to work on it. If you haven't taken exam 2, I need you to get on it and take it. If you go in, and I know that not all the scores always transfer over, and sometimes D2L, Connect doesn't even give you a score, but you, on your own records that Connect gives you, have a score for it. Courtney, you and I have had this problem before, and I think we might be having the same problem again. And she's probably not the only one. If you have a score for the exam, I need, and you don't, it's not recorded in D2L. I need you to send me a screenshot with your score, because it means that I don't have it and I've never seen it. So send me.
Sorry, I was just saying, thankfully, I actually checked that when I got on today, and it's in D2O.
Is it? Okay.
Yes.
Okay, good. All right, then we won't worry about you. Anyone else, please get it. Please make sure that I have a score for you. And that really goes without saying for any of the Connect assignments or any of the Connect assignments. If you go into D2L and you do not have a score, it means that Connect my Connect record doesn't have a score for you either. Connect is great, but every once in a while there's a little glitch in the system. And it just means that we both have to make sure that your grades are all in. All right, let's get started. I think we were about right here, maybe. Like all reflexes and feedback loops, visceral reflexes have 5 components. What are they?
Could be receptors.
Yep, the receptors, the stimulus and the receptors.
Yeah, afferent is sensory receptor sending to central.
Mm-hmm.
Then efferent and effector.
Correct. So sensory neurons carry the nerve impulses from the stimulated sensory receptors. In the visceral organs, we're talking about visceral reflexes. Same components, different situation. And blood vessels to the central nervous system for integration. Autonomic motor neurons carry nerve impulses from the central nervous system to smooth muscle, cardiac muscles, and glands and effectors. An example of a visceral reflex arc is the Biol reflex. Have you heard of a barometer before?
Yes,
What is a barometer?
for pressure, atmosphere pressure.
It's to measure pressure. So a Biol reflex is a reflex against too much pressure. So we have baroreceptors that sense the levels of pressure in the vessels, in the blood vessels. And the internal, they're in the internal, let's see here, bear with me here, I just gotta make sure I'm doing this right. I think they're in the common carotid artery. It says the internal, I'll go with the internal carotid artery and the aortic arch. The glossopharyngeal nerve sends a signal. What cranial nerve is the glossopharyngeal nerve? Someone besides Layla. What one is the glossopharyngeal nerve? Which cranial nerve is it? Find a mnemonic that you use. What cranial nerve is it? What nerve is the glossopharyngeal nerve? What cranial nerve?
Is it number 9?
It is number 9. Good job. It is number 9. So glossopharyngeal nerve sends a signal to the medulla oblongata. If you remember, the medulla oblongata is what contains the cardiovascular centers and the breathing rhythmic center. Signal goes up to the medulla oblongata, integrating center there. Vagus nerve, which cranial nerve is that? Which one is the vagus nerve?
Is that number 10?
It's number 10, correct. So the vagus nerve transmits an inhibitory signal back to the cardiac pacemakers. Next semester, you're going to learn a lot about the heart. But in the right atria, there are what we call the cardiac pacemaker, the sinoatrial node. And it sends a signal. It regulates the beating of the heart. So the vagus nerve sends an inhibitory signal to the cardiac pacemaker, decreasing the heart rate. And from that, the blood pressure is reduced. The less blood that you're pumping out into those vessels, because the heart rate has gone down a little bit, the less pressure there is going to be there. So this is an example of a homeostatic negative feedback loop. Just like all our other reflex arcs, they are homeostatic feedback loops. I think we were further along than this, but we'll keep going from right here. Dual innervation. A lot of the visceral organs and a lot of anything regulated by the autonomic nervous system, which is almost all of it. I don't know, outside of skeletal muscle, which is sometimes regulated by the autonomic nervous system. Most things are regulated by the autonomic nervous system. And most of them have dual innervation, where the sympathetic and the parasympathetic divisions innervate many of the same organs. They can have contrasting effects or cooperative effects. Contrasting meaning they have opposite effects, or cooperating meaning they enhance the activity in different ways. And they build on the activity in different ways. Different effects are due to the different types of neurotransmitters and the different types of receptors on those effectors. One speeds things up, one slows things down. So, for example, parasympathetic fibers of the oculomotor nerve, cranial nerve number 3, pass through the ciliary ganglion. Remember, this is going to be where synapse is going to occur. Ciliary ganglion. And they have a cholinergic stimulation of the pupillary constrictor, pupillary, as I say it all wrong, pupillary constrictor, relaxing that muscle. When you relax that muscle, the iris is a muscle in itself. When the muscle shortens, it's contracting. When it's relaxed, it's lengthening out. So in this case, it's lengthening out. It's relaxing. So the parasympathetic fibers relax the muscle of that makes up the iris. So parasympathetic leads to a constriction of the pupil. Relaxation of the iris muscles leading to a constriction of the pupil because the pupil becomes smaller. Even though the muscle is relaxing, the pupil is getting smaller. Just based on the length of the muscle here. Is it contracting or is it relaxing? Sympathetic fibers. Superior cervical ganglion. Now, has an adrenergic stimulation. We're going to talk about the difference between cholinergic and adrenergic in the next couple slides. Adrenergic stimulation causing constriction of the iris muscle, causing the pupil to dilate. The iris muscle, can see the length difference here. Goes from being like 2 millimeters here or 3 millimeters here. Let's say it goes from being like 4 millimeters here to 2 millimeters here. So it's contracting because it's getting stimulated by the sympathetic nervous system to contract. Two major subdivisions of the autonomic nervous system. We have the sympathetic nervous system, which is your fight or flight. You saw me when I ticked off my cat. I can stimulate his sympathetic nervous system fairly easily by just getting him mad. Prepares the body for intense physical activity, concerned with processes that expend energy. Parasympathetic nervous system, rest and digest. Relaxes the body and inhibits or slows many high energy using functions. Concerned with activities that restore or conserve energy. Sympathetic response stimulated by the four E's. An emergency, excitement, or another one that we could say this right here. Well, maybe not. I would say even emotion, but usually excitement is an emotion, embarrassment is an emotion, and exercise. Your sympathetic nervous system causes your heart rate to increase, your liver to release glucose into the blood that you can use for energy. Bronchioles of the lungs dilate, pupils dilate. The adrenal glands secrete epinephrine and norepinephrine, hormones which give the same but longer lasting effects than those of the sympathetic nervous system neurons. Going to talk about how they're unique, the adrenal glands, actually specifically the adrenal medulla. Digestive activities are inhibited and the bladder relaxes. For example, the brain recognizes a bear as a threat and unconsciously activates the sympathetic nervous system. When the threat's over, the peripheral nervous system is unconsciously activated. The body calms so that it can recover from the heightened physical activity and promote restoration and repair. It takes longer for your parasympathetic nervous system to be activated than for your sympathetic nervous system to be activated. And you can understand why that would be important. Your body is going to protect you from threats, and so your ability to become more activated a lot faster is better than your body relaxing a lot faster. Heart rate decreases, liver no longer releases glucose and produces bile, which is going to absorb nutrient fat, help with the fat absorption. Bronchioles of the lungs constrict, pupils constrict. Adrenal glands stop secreting epinephrine and norepinephrine. Digestive activities are stimulated, the bladder contracts. Which of the following effects are mediated by the sympathetic nervous system? Bladder contraction, inhibition of the adrenal glands, increased heart rate, pupil constriction, decreased lung capacity.
Would it be C and E?
I don't think it's, let me think. It's not E. And the reason is because increased lung capacity is sympathetic nervous system. It's when their bronchioles dilate.
Oh, because I thought whenever you're like in the sympathetic, you take short breaths, and that's why it's not.
The C, increase heart rate.
I would say it's C. I don't know about the short breaths. I'm trying to think.
Because when you're relaxed, you take like longer, deeper breaths. But when you're like excited or doing something, like your breaths are shorter and
Your heart rate goes up, and that causes you to take breaths a little faster, I think. But I don't know if it has to do with if that's your short breaths are an indirect response to your heart rate going up.
I think breath will be increased because of heart rate need more oxygen. It will be more breath rate. It
Yeah,
will be shorter, but it is faster as you're running or something like this.
yeah. I think the short breaths are, I think that is an indirect response of other things. I don't know. Don't know about that one. Yeah, let me think on that one a little bit. But it is sorry, what was the right answer? It was C. The increased lung capacity is the sympathetic nervous system. I need to think about that short breaths. Yeah, I need to think about that one. Which of the following effects are mediated by the parasympathetic nervous system? Stimulation of the digestive system, secretion of epinephrine by adrenal glands, increased blood flow to the lungs, release of glucose into the bloodstream by liver or bronchial dilation.
It's A.
It's A. Yeah, it's A. It's stimulation of the digestive system because it's the rest and relax where your body's actually recovering from a strong energy expenditure and allowing you to start storing energy again.
Actually, the sympathetic is increased dilatation of bronchus and increased blood flow to the lung. It means increase the breathing, actually.
Say it one more time.
It is increased breathing because parasympathetic, it is increased dilatation of bronchial and increased flow to the lung. So it means increase the breathing rate, capacity of the lung, increase the capacity of the lung, not decrease.
Mm-hmm. The parasympathetic and parasympathetic nervous system are two divisions of which other nervous system? Let's go with E, the autonomic nervous system. So the autonomic nervous system has components in the central nervous system and the peripheral nervous system. There are control nuclei in the hypothalamus. Remember that the hypothalamus is the main regulator of the autonomic nervous system. And other brainstem regions, such as the medulla oblongata, where your cardiovascular and your breathing rhythmicity centers are. It also has motor neurons in the spinal cord and peripheral ganglia. Spinal fibers travel through the cranial and spinal nerves. Stretch receptors, mechanical receptors, irritant receptors, nociceptors. A lot of them are nociceptors. meaning pain. Receptors in the joints and in the muscles, your spindle fibers, your tendon organs. Chemoreceptors, sense the increased oxygen levels or the decreased oxygen levels, increased carbon dioxide levels, increased amounts of hydrogen ions. One thing with regards to the question earlier about the heart rate going up, you can increase your heart rate. Increasing your breathing rate can cause your blood to become more, I have to remember how this works, cause your blood to become more acidic. And the reason that breathing in carbon dioxide is because it brings, the reason breathing in a bag will bring down your breathing rate is because it changes the acidity. And I would just have to think about how that sympathetic nervous system works with increased heart rate. But part of that has to do with the fact that your blood pH is dropping. And that'll be another thing for next semester. Comparisons of the somatic and the autonomic efferent pathways. Somatic nervous system effectors are skeletal muscles, and the effect is always excitatory. The efferent fiber is, with regards to the somatic nervous system, is myelinated, and it releases acetylcholine as the neurotransmitter. In the case of the autonomic nervous system, there are two neurons from the central nervous system to the effector, so it's not just one. There's the myelinated preganglionic, so before the ganglion, autonomic ganglion where there's a synapse. You remember we saw these up here. Of this, here's the preganglionic neuron, here's the postganglionic neuron. So past the ganglion, kind of like the pre and post central gyri of the brain. The preganglionic neuron, the postganglionic neuron. So preganglionic neuron, postganglionic neuron. Remember, nerve fiber, neuron, think of them as being the same thing here. The neuron versus the nerve fiber. In the case of the postganglionic neuron, it is unmyelinated. Autonomic nervous system, the postganglionic neuron can secrete acetylcholine or norepinephrine. The preganglionic neuron secretes acetylcholine. Remember that acetylcholine is one of the neurotransmitters. Norepinephrine is another one of the neurotransmitters. Autonomic nervous system effectors are cardiac muscle, smooth muscle, and glands. And the effect can either be excitatory or inhibitory. So comparing and contrasting a little bit here. Effectors, skeletal muscle for somatic. For the autonomic nervous system, glands, smooth muscle, cardiac muscle. Control, somatic, usually voluntary. Autonomic, usually involuntary. Distal nerve endings, neuromuscular junctions, which we'll talk about in the next chapter. Remember, we're covering chapter 11 first, and then we will go back and cover the sense organs. Autonomic nervous system, distal nerve endings of varicosities. Efferent pathways for somatic nervous system. One nerve fiber from the central nervous system to the effector, and there's no ganglia. The autonomic nervous system, there's two nerve fibers from the central nervous system to the effector. And there's a synapse in the ganglion. Neurotransmitters for is acetylcholine in the somatic. Neurotransmitters in the autonomic nervous system are acetylcholine and norepinephrine. Effect on the target cells. Automatic, always excitatory. Autonomic nervous system, excitatory or inhibitory. Something that we won't spend a lot of time on, but in the case of denervation, let's say denervation, flaccid paralysis of that muscle. Doesn't get it relaxes and it can't contract. Denervation hypersensitivity becomes overly sensitive and overly contracted. It kind of shakes. So autonomic nervous system, preganglionic neuron or nerve fiber, myelinated. Acetylcholine in the autonomic ganglion. At the effector site, acetylcholine or norepinephrine. Postganglionic neuron is unmyelinated. Smooth muscle contraction or relaxation. Cardiac muscle can either be increased or decrease the rate and force of contraction. Glands have increased or decreased secretions. Autonomic postganglionic fibers have faster or slower conduction speeds than somatic motor fibers. What do you think? Would autonomic postganglionic fibers have faster or slower conduction speeds than somatic motor fibers? Here's the somatic, here's the autonomic. What do you think? Somebody tell me what they think. Would autonomic postganglionic fibers have faster or slower conduction speeds than somatic motor fibers?
He has both actually.
No, it doesn't. Why do you think it has both?
Because he said it is can excite or inhibitory. Excitatory or
Well,
inhibitory.
what about conduction speeds through the fiber?
It will be slow because unmyelinated.
Exactly. Yes, you're right. It could inhibit or excite it, but the conduction speed through the fiber is going to be slower because unmyelinated. You're right, Leila. So preganglionic versus postganglionic motor neuron. So remember, this is before the ganglion, this is after the ganglion. The preganglionic neuron is the first of two autonomic motor neurons. The cell body is in the brain or the spinal cord. The myelinated axon passes out of the central nervous system as part of the cranial nerve or spinal nerve and extends to an autonomic ganglion. And synapses with a postganglionic neuron that is unmyelinated. For the postganglionic neuron, it is the second of two autonomic motor neurons. It lies entirely outside of the central nervous system. The cell body and the dendrites are located in the autonomic ganglion. It synapses with one or more preganglionic neurons, so it can synapse with multiple ones here. And the axon of the postganglionic neuron is unmyelinated. And the axon of this one terminates at the visceral effector. So differences, first of two, cell body is in the brain or spinal cord. Myelinated preganglionic fiber passes out as part of the cranial nerve or spinal nerve, extends to the autonomic ganglion, synapses with a postganglionic neuron that's unmyelinated. Postganglionic neuron, second of two autonomic motor neurons, lies entirely outside of the central nervous system. And the cell body and dendrites are located in the autonomic ganglion. It synapses there with one or more. So here we only see one, but it can be more than one. Preganglionic fibers. And the axon of the preganglionic fiber is unmyelinated. The axon terminates at a visceral effector. Sympathetic nervous system, also sometimes called the thoracocolumbar division, because it comes off the thoracic spinal nerves and the lumbar spinal nerves. The preganglionic neurosomas are located in the lateral gray horns. Do you remember? Actually, it's in the next question here. Where do you see the lateral gray horns along the spinal cord?
Gray matter?
Well, where do you see them? Where do you see them, Layla? Where do you see the lateral gray horns along the spinal cord? You don't see them throughout. Where do you see them? You see them between the first thoracic vertebrae to L2, the second lumbar vertebrae. Because the lateral gray horn contains the preganglionic cell bodies for the sympathetic nervous system. That is why the sympathetic nervous system is frequently also called the thoracolumbar division, is because of where the lateral gray horns are present, which have the cell bodies of the preganglionic neurons for the sympathetic nervous system. Remember, we talked about the lateral horns of the spinal cord. So this is where the cell bodies for the preganglionic cell This is the lateral horns are where the preganglionic cell bodies are, or neurosomas. They lead to the nearby sympathetic chain ganglia, or paravertebral. Paravertebral meaning para, meaning next to or by the side of, vertebral. So by the side of the vertebral column, vertebrae. Sympathetic nerve fibers are distributed to every level of the body. They have relatively short preganglionic fibers and long postganglionic fibers. They're relatively short because most of them will synapse about right here. They don't extend very far. Remember, this is the paravertebral, so it lines up almost right against the vertebral column. The paravertebral ganglion, look here, here's the vertebral column, here's the paravertebral ganglion where a lot of the presynaptic ganglion synapse. Each paravertebral ganglion is connected to an autonomic nerve by two branches. The branches of the rami communicante. You remember that word when we were talking about spinal cord anatomy? The preganglionic fibers are small myelinated fibers that travel from the spinal nerve to the ganglion via the white communicating ramus. And remember, every time we talk about something being white in the nervous system, we're referring to the fact that it's myelinated. Because when it's not myelinated, it looks gray. The postganglionic fibers leave the ganglion by way of the gray communicating ramus, the unmyelinated fibers. And they extend the rest of the way to the target organ. I'm just going to come back here really quickly. So, preganglionic neurons of the sympathetic nervous system come off from T1 to L2, those spinal nerves. They extend out, and they don't extend out very far. They just extend out to the sympathetic ganglion. or the paravertebral ganglion? So they have relatively short preganglionic fibers and long postganglionic fibers. The preganglionic fibers are small myelinated fibers that travel from the spinal cord to the ganglion by way of the white communicating ramus. The postganglionic fibers leave the ganglion by way of the gray communicating ramus. The white rami communicantes carry the preganglionic sympathetic fibers from the spinal nerves to the sympathetic ganglia. Most of those are in the sympathetic chain ganglia or the paravertebral ganglia. The gray rami communicantes carry the postganglionic sympathetic fibers from the sympathetic ganglion to the spinal nerves. That isn't right. Let's say some, oh, it doesn't look completely right to me. I need to think about that one. Now that I put it in there, it doesn't look completely right to me. I guess it could be right. Postganglionic efferent. Yeah, let me think about that. But go with this up here. So actually, I'm going to keep going and then I'll come back and review. After, yeah, actually, let's stop about right here. Are there any questions? Are there any questions?
No.
Let's keep going for a little bit. After entering the sympathetic chain ganglion, remember these preganglionic fibers usually don't tend to be very long because they don't go very far. Most of them will connect with the postganglionic neuron within the sympathetic chain ganglia, which is also called the paravertebral ganglia because it's right next to the vertebral column. So they don't have to be very long. So remember that sympathetic nervous system tends to have very short preganglionic fibers. So it can follow one of three courses once it enters the sympathetic chain. They can either enter a ganglia and synapse immediately with a postganglionic neuron. Right here. And from there, they go out to sweat glands, the erector muscles, and blood vessels of the skin and skeletal muscles. Or they can travel up or down the spinal cord and synapse in a ganglia at other levels along this chain. And for that postganglionic, those postganglionic neurons and go out to irises, salivary glands, lungs, heart, thoracic blood vessels, or the esophagus. Third thing that they can do, once they enter the preganglionic chain, they can pass through this chain without synapsing and continue as a splanchnic nerve that leads to a second set of ganglia called the collateral ganglia that go out to the liver, the spleen, the adrenal glands, the stomach, the intestines, the kidneys, the urinary bladder, the reproductive organs. So it 3 different courses here. It can enter the chain. These preganglionic neurons of the sympathetic nervous system can enter the chain, immediately synapse with a postganglionic neuron, and those postganglionic neurons lead to sweat glands, the piloerector muscles, the blood vessels of the skin and skeletal muscles. Or they can enter the chain and travel up or down and synapse and ganglia at other levels along the chain. Postganglionic neurons that connect with these preganglionic neurons go out to the iris, the salivary glands, the lungs, the heart, the thoracic blood vessels, and the esophagus. Third, they can pass through the chain without synapsing at all and continue as a splanchnic nerve that leads to a second set of ganglia called the collateral ganglia. Remember collateral? You remember collateral axon? Kind of an off branch? It's the same thing here, it's an off branch ganglia. Collateral ganglia that are we know they're part of the sympathetic nervous system, or prevertebral ganglia, contribute to a network called the abdominal aorta plexus. Plexus meaning a branch, inner network branch, inner network of neurons. And it's the abdominal aorta because it wraps around the abdominal aorta. The aorta is given different names depending on where it is along your trunk. Thoracic aorta up here, abdominal aorta down here. And many postganglionic fibers exit this plexus. So they branch around it, they chain and branch around the abdominal aorta, and they exit this plexus and accompany arteries with the same name and follow the same routes to their target organs. So for example, here's the superior mesenteric artery. There's a branch of the collateral ganglia that is called the superior mesenteric nerve. And it's part of this collateral ganglia that it branches off here. We have an inferior mesenteric ganglia. The celiac ganglia. So the collateral ganglia contribute to the abdominal aortic plexus. Sympathetic ganglia that belong to the collateral ganglia. Thoracic ganglia 5 through to 9 or 10 are part of the celiac ganglion that lead out to the stomach. They're effectors of the stomach, the spleen, the liver, the pancreas, small intestines, and kidneys. Thoracic ganglia 9 through 12 lead out to the celiac ganglia and the superior mesenteric ganglia. They're effectors of the small intestines, the colon, and the kidneys. The lumbar ganglia are the lead out to the inferior mesenteric ganglia, rectum, urinary bladder, and reproductive organs. Guys, why do we just have them in the thoracic and the lumbar region? Why don't we have them in the cervical area and the sacral region? Right.
Because of lateral horn.
What?
Lateral horn only in this region.
Right. Well, yeah, it's only in the lateral horn region, which is T2 through T1 through L2. And the sympathetic nervous system is only comes off the thoracic through the lumbar region, like Leila said, because it only comes off the lateral horn. Sympathetic innervation to the adrenal medulla. You remember where the adrenal glands are, right? Where are the adrenal glands?
The gland at the top of kidney.
Yep, they're the glands above the kidney. Adrenal, renal meaning kidneys. Medulla, remember the medullary cavity of the bone? Where was that located? Where was the medullary cavity of the bone? Guys, where was the medullary cavity of the bones? Bringing this up for a reason. Where was the medullary cavity of bones?
The long bone.
Where was it in the long bone?
In the middle of the bone.
The middle. It's in the inner side. It's in the interior. So the adrenal medulla is the interior cavity of the adrenal glands. Medulla meaning cavity, inside, interior cavity. Pre-ganglionic sympathetic fibers do not synapse in the sympathetic chain ganglion. Remember, these are ones that are going to pass through. They go directly to the adrenal medulla and release acetylcholine into chromaffin cells, which are modified. Chromaffin cells that are part of the adrenal medulla are modified postganglionic neurons. They do not contain axons and they do not contain dendrites. They're just cell bodies. That's the reason they're modified. So they aren't, they're similar to the collateral ganglia in that they don't synapse. There's these postganglionic neurons that go out to the adrenal medulla do not synapse in the chain. They continue like the splanchnic nerves do that are going to go out to the collateral ganglia. But instead of going to the collateral ganglia, they go out to the adrenal medulla. And the chromaffin cells release epinephrine, also called adrenaline, and norepinephrine or noradrenaline. It's called adrenaline because it's secreted by the adrenal gland. So these preganglionic fibers do not synapse in the chain. But they go out and synapse in the adrenal medulla with the chromaffin cells, which are modified postganglionic neurons. And here they secrete acetylcholine onto these neurons that then release epinephrine and norepinephrine. Preganglionic sympathetic fibers exit the spinal cord via the blank, pass through the blank to enter the what?
I'm guessing it's DOP.
I would say it's B.
Okay.
And if you follow here, ventral root, anterior root,
Let's see.
where were we? Rami communicantes, sympathetic chain ganglia. The primary reason the sympathetic division of the autonomic nervous system produces immediate responses within the body is based on the concept of divergence. We didn't really talk about this. I think The answer is false. I wouldn't ask you this before. I think your book says it specifically, but I think that it's not a divergence, it's a convergence process. If you remember, I'm going to come up here really quick. Bear with me just for a second here. Synapses with one or more, it's convergence, it's not a divergence. Synapses with one or more preganglionic fibers, meaning that multiple preganglionic fibers can converge on one postganglionic fiber. So it's not a divergence, it's a convergence. So that's why that answer is false. Parasympathetic. Okay, so I'm gonna just do a quick review here. Come back up here. Sympathetic division comes off the lateral horns, which are only present from T1 to L2, which is the thoracolumbar division. They have short preganglionic fibers and long postganglionic neurons. Most of them enter the sympathetic chain ganglia and synapse within there with a postganglionic fiber. Some will pass through and become part of the splanchnic nerves or go out and innervate the adrenal medulla. In contrast, within the parasympathetic division, the parasympathetic nervous system comes out of the cranial part of the spinal cord and the sacral part of the spinal cord. And the fibers travel within certain cranial and sacral nerves. So sympathetic nervous system comes out of this area of the spinal cord. Parasympathetic nervous system comes out the ends, either the cranial end or the sacral end. Instead of having short preganglionic fibers like that seen in the sympathetic nervous system, they have long preganglionic fibers whose axons end in terminal ganglia in or near the target organs. With the ganglia being so close to the target organs or the effector organs, they're going to have short postganglionic fibers. So sympathetic nervous system is short preganglionic fibers that come off the thoracolumbar region within the lateral horns. They have long postganglionic neurons. Parasympathetic comes off the cranial or sacral regions of the central nervous system. And they have long preganglionic fibers and short postganglionic fibers. There is little neural divergence. Actually, I may have been wrong about this one here. This I think is true. Because I think the difference is that there is divergence in the sympathetic nervous system, and there's convergence in the parasympathetic nervous system. I'm obviously not going to bring that up with you again. And I'm not going to ask you about it on an exam if I can't get it straight in my head. But the parasympathetic division doesn't have as much divergence as the sympathetic nervous system has. So remember, this is the craniosacral division. So a lot of the cranial outflow of the parasympathetic nervous system is from the cranial nerves. The oculomotor nerve, cranial nerve number 3, that narrows the pupil and focuses the lens. The facial nerve, which allows for tears, nasal, mucus, and salivary glands. The glossopharyngeal nerve, the parotid salivary glands. And the vagus nerve, which is many of the viscera. The remaining parasympathetic fibers arise from the sacral nerve number 2 to sacral nerve number 4 of the spinal cord and form the pelvic splanchnic nerves that lead to the inferior hypogastric, so underneath the stomach, inferior hypogastric plexus. So sympathetic nervous system comes off the thoracolumbar region of the spinal cord. The cell bodies are in the lateral horn. They have short preganglionic neurons, long postganglionic neurons. And what else was there? I think that was it for that. And parasympathetic comes off the cranial area and the sacral area. They have long preganglionic neurons and shorter postganglionic neurons. Origin in the central nervous system, sympathetic, thoracolumbar region. Parasympathetic, craniosacral region. Location of ganglia. Paravertebral ganglia adjacent to the spinal column and prevertebral ganglia anterior to it. Parasympathetic, terminal ganglia near or within target organs. Fiber lengths, short preganglionic, long preganglionic. Long postganglionic, short postganglionic. Sympathetic, there's extensive divergence. Parasympathetic, there's minimal divergence. Effects of the nervous system, often widespread and general. Effects of this nervous system, more specific and local. There's very little divergence. It's going to be much more local. If it's not going to diverge out in multiple ways, it's going to be more local. Enteric nervous system does not arise from the brain or the spinal cord. Whether or not, it's often debatable whether we call this, and if we consider this as being part of the autonomic nervous system or not. However, because it is not voluntarily controlled, it's easier to put it with the autonomic nervous system than the somatic motor nervous system. Composed of 1 million neurons found in the walls of the digestive tract. I'm just writing here. This is the GI tube, gastrointestinal tube, which is essentially one tube from your mouth to your anus. It includes the myenteric plexus and the submucosal plexus. Myenteric plexus allows for GI tract motility, peristalsis. Submucosal plexus controls secretions of glands into the GI tract. Enteric nervous system, submucosal and myenteric. Ileum vasculature, muscle. How do autonomic neurons have, bear with me, how do autonomic neurons have contrasting effects on the same organs? Actually, you guys, let's stop right there for 10 minutes. Let's come back at 9:35, okay? If you're anything like me right now, you're tired of sitting in one place. Let's come right back to here. We will finish this out today, and we will start the muscle, chapter 11. So get up and move around for a minute, and I'll see you back here at 9:35.
Hey, Professor, I had a quick question.
Go for it.
On the Lab 8 worksheets, I'm behind on my worksheets. I know. I'm trying to get them done. But on the beginning part of it, it says to use the pictures on the actual PowerPoint to basically label, like to say which part of the yeah, I'm having a hard time today. Basically the skull. Let me just bring it up because now I'm like blubbering.
Well, let me go into it too. Here, why don't I look at it? Let's see here. I understand the having a hard time thing.
My son woke up, he threw me off, and I'm like, wait, what am I doing again?
Yeah, I didn't see a score for your exam, so I'm glad if it's on there, it's on there. Then I'm not worried about it. But I'll look for it again later today.
Yeah, and I can double check in, but I'm pretty sure because it said 95 out of 100. So.
All right, let's see here. Okay, so lab content.
Wake up, Mickey! We go house! Mickey, this is a cutter!
Yeah.
Got to keep you busy.
Yes.
Very important.
Definitely does.
So the axe is lab 8.
Yes. Yeah, the lab.
Skeleton.
Yes.
Okay. worksheet. Okay.
Shh, hold on, Bubba.
I don't think you. Okay, go ahead. Tell me where it was.
It was in the very first part. It says, oh wait.
Use the pictures to. Okay, use the pictures to identify all of the. Okay. You don't have. That's not a question. There's just a few questions in here, like define a suture.
Okay.
There's only a handful of questions in here.
Okay. I wanted to make sure because I was looking back on the lab and I'm like, okay, is there
No,
basically like
no, there's just a handful of questions. Define a suture. That's from, I think that's the first one there. What is a cleft lip and palate? What do we commonly call a fontanelle? What is their function?
okay.
This part right here,
Okay.
this right here. Depending on how much chaos your son gives you, you could probably have this done within about 20 minutes.
Okay.
Define the abnormal curvatures. That's it.
Okay. All right. I just wanted to make sure because I'm like, okay, I didn't know if there was a separate like worksheet from the lab or okay.
That's all there is.
All right, awesome. Thank you.
You're welcome. Any questions or thoughts about the autonomic nervous system?
Just more complexities to try to understand.
Don't say that. Don't say that. Don't put it like. Don't put it like that. There's certain things that simplify it down. Like I always try to remember parasympathetic. If I think of it as the parasympathetic, para meaning it comes off the sides of the spinal cord, so it comes off the ends of the spinal cord. So it comes off the end, and it has longer preganglionic fibers. It has shorter postganglionic fibers, neurons. Sympathetic comes off the middle, the thoracolumbar region, from the lateral horns. It has short preganglionic fibers and long postganglionic fibers. How do they have opposite contrasting? How can they have contrasting effects on the same organs? It's because they secrete, one reason is because they secrete different neurotransmitters that have different effects. Norepinephrine and acetylcholine. Moreover, in addition to the fact that they secrete different neurotransmitters, they have, based on their receptors that can vary significantly, the target cells can respond to the same neurotransmitter differently, depending on which receptor that neurotransmitter is binding. Whether that receptor is present or not will depend on the activities that neurotransmitter will activate. Despite the variety of possible chemicals that could be used to submit chemical messages in the ANS, the autonomic nervous system, acetylcholine, norepinephrine, and epinephrine are used in the majority of the times, are used to any great degree. There are well over 100 neurotransmitters in the body. And the autonomic nervous system tends to use acetylcholine, norepinephrine, and epinephrine more than any of the others. If you look up here, here's acetylcholine, here's norepinephrine, here's epinephrine. Cholinergic neurons are synapses, acetylcholine, cholinergic. Cholinergic neurons are synapses at which acetylcholine is used, and these are termed to be cholinergic. They tend to be nicotinic receptors or muscarinic receptors. So receptors for acetylcholine are nicotinic receptors or muscarinic receptors. There are the synapses for all preganglionic neurons in both divisions. And that let's see here. That doesn't really make sense to me. There are, it made sense to me when I wrote it in there, but now I'm looking at it and I'm having to think through it. There are synapses for all preganglionic neurons in both divisions. And that of all postganglionic parasympathetic neurons. I need to think through that one. Remember that cholinergic receptors are nicotinic receptors and muscarinic receptors. Any receptor that binds acetylcholine is a cholinergic receptor. And there are two types. Nicotinic, the two types of the nicotinic receptor and the muscarinic receptors. Muscarinic receptors are found on all cardiac muscle, smooth muscle, and gland cells. They can be excitatory or inhibitory due to the subclasses of muscarinic receptors. They are named, they're named muscarinic receptors because the toxin muscarine, which comes from a poisonous mushroom, irreversibly binds to this receptor and blocks acetylcholine's activities. So it is found, muscarinic receptors are found on the plasma membrane of all effectors, smooth muscle, cardiac muscle, and glands that are innervated by the parasympathetic postganglionic axons. Muscarinic receptors are found on the plasma membranes of cells of most sweat glands, innervated by sympathetic postganglionic neurons. nicotinic receptors. Can you guess why they're called nicotinic receptors? What do you know of that's probably one of the most addictive drugs out there that is legal today?
When you say addictive drugs, do you mean like for pain, for?
It doesn't matter. Pain, anything. The most legal drug that you know out there that is it is one of the most addictive drugs out there, but it's also very legal.
Oxy?
Nope.
Nicotine?
Nicotine.
Mm-hmm.
It's nicotine. Oxy is actually regulated. Nicotine's a little regulated, but not to the extent that oxy is.
Okay.
It binds to these receptors and blocks acetylcholine.
Now, can that be different for different people? Because I was more of like a social smoker, or like when I drank, but I usually like cigars, but like.
You're just weird.
I am.
Do it.
I had a pack of cigarettes for a year, and my friend's like, "Can I bum one off you?" I'm like, "Yeah, sure." And she's like, "These taste terrible. How long have you had them?" I said, "A year." She's like, "Ew, they're expired." I'm like, "They taste fine to me."
Well, if you get really nasty cigars, that's why you're never gonna get addicted. Maybe that's how to avoid addiction.
Maybe.
You know what? I have a friend, and I told you about her before, that had the Chiari malformation.
Yeah.
She also has an issue where she does not become easily addicted to anything. And her issue is that she's a weirdo like you. She's probably a little bit more weird than you are. Her issue is that she does not secrete any serotonin. She's been tested, and she has no production of serotonin.
Oh, wow.
And serotonin is one of the ways that addictions become so effective. A lot of our neurotransmitters and our autonomic nervous system is how drugs tend to work. Okay. And the fact that she does not make any serotonin makes her so that she has a very unaddictive personality. She just doesn't become addicted to anything. And she's just a weirdo. But she knows it. So nicotinic receptors, the reason nicotine is it's next to heroin in its potency for addiction. Nicotinic receptors are always at the end of the autonomic postganglionic neurons within the adrenal medulla, and are excitatory when acetylcholine binds. It's always going to result in the release of epinephrine or epinephrine. So muscarinic receptors found on the plasma membrane of smooth muscle, cardiac muscle, and glands, innervated by the parasympathetic postganglionic axons. Bear with me, I'm sorry. Muscarinic receptors are also found on sweat glands, innervated by sympathetic postganglionic neurons. Nicotinic receptors, always found at the end of postganglionic neurons within the adrenal medulla, and are excitatory. So they're always going to result in the secretion. Nicotinic receptors are also found at the end of postganglionic neurons and somatic motor neurons. Going to talk about this in our next chapter, chapter 11, at neuromuscular junctions with skeletal muscle. are always excitatory when acetylcholine binds. I think it's really interesting that a lot of people who smoke or use nicotine use it to relax, but it's always excitatory in its effect. So at the end of postganglionic neurons and somatic motor neurons at neuromuscular junctions with skeletal muscle, and is excitatory when acetylcholine binds. Actually, I think I said that wrong. It blocks the effects of acetylcholine. So it is going to be a relaxant majority of the time, most of the time. Adrenergic neurons, synapses at which norepinephrine and epinephrine. Adrenergic meaning adrenaline or noradrenaline. Adrenaline, noradrenaline. Adrenergic neurons. So that's where the name is coming from, is because these were once called adrenaline and noradrenaline because of where they come from, the adrenal medulla. So synapses at which norepinephrine and epinephrine are used are termed to be adrenergic. They are at the synapses for nearly all sympathetic postganglionic neurons. I think what I made a typo, and that's why I'm looking at this thinking, Mary, what were you thinking? They are at the synapses for all preganglionic, so cholinergic neurons receptors are at the synapses for all preganglionic neurons in both divisions. So acetylcholine, that's where I've got a typo here. So they are in the synapses, at the synapses for all preganglionic neurons in both divisions, and those of all postganglionic parasympathetic neurons. So it's created. So this is just saying where the receptors are found. Cholinergic receptors are at the synapses of all preganglionic neurons in both the both sympathetic and parasympathetic nervous systems. And it is found at all of the postganglionic parasympathetic neurons. Adrenergic receptors are found at nearly all of the synapses for sympathetic postganglionic neurons. So when I said that it didn't really make sense to me, it's because there's a typo here. They are at this, or it is found at the synapses for nearly all sympathetic postganglionic neurons. There are two types of adrenergic receptors. There's actually two subclasses, two types, and then two subclasses for each type. Any receptor that binds epinephrine or norepinephrine is called an adrenergic receptor. Remember, it's because they're also called adrenaline and noradrenaline. Alpha adrenergic receptors have two subclasses where alpha 1s are usually excitatory and alpha 2s are usually inhibitory receptors. two subclasses of beta adrenergic receptors. A beta 1 and a beta 2. With different effects. Sympathetic nervous system. All preganglionic fibers are going to secrete acetylcholine. Most preganglionic fibers, most postganglionic fibers of the sympathetic nervous system are adrenergic, with a few of them being cholinergic. Adrenergic, meaning they secrete acetylcholine. I can't believe I just said that. Mostly adrenergic, meaning they secrete norepinephrine or epinephrine. A few are cholinergic, meaning that they secrete acetylcholine. Parasympathetic, always cholinergic, and ganglionic fibers. Always cholinergic. So most adrenergic receptors are going to have to do with the sympathetic nervous system. Actually, almost all of them are going to have to do with adrenergic, with the sympathetic. Almost all adrenergic receptors are going to be with regards to the sympathetic nervous system. All others are going to be cholinergic, with a few. Okay. I'm trying to narrow things down and classify them a little bit for you. Sympathetic is always the ganglions between the preganglionic fibers and the postganglionic fibers are always going to be cholinergic synapses, where acetylcholine is released. Postganglionic, mostly adrenergic, meaning that it's mostly secretes norepinephrine and epinephrine. And there's a few cholinergic, meaning some secrete acetylcholine, but not very many. Parasympathetic postganglionic fibers always secrete acetylcholine. Most drugs, like nicotine, which isn't, since it acts on our own human cells because it's targeted to them, they generally act by binding to proteins like ion channels, enzymes, receptors, and carrier proteins. The endogenous ligand, the normal ligand for a receptor, binds to the receptor and produces an effect. An agonist binds to that receptor and produces the same effect as the endogenous natural ligand would. An antagonist is close enough in shape to the natural ligand, but when it binds to that receptor, it blocks the effect and prevents the natural ligand from binding. So an agonist stimulates the same effect that the natural ligand would produce. An antagonist blocks the receptor, doesn't allow the natural ligand to bind, and doesn't allow activation of the receptor. Other drugs, for example, antibiotics, are targeted at the proteins of pathogens. Reason that antibiotics generally don't hurt you is because they don't bind to your body's proteins or block the activities of any of your proteins. Bind and or block the activities of any of your proteins. They're designed to go after the proteins specific to bacteria. An example of a drug are beta blockers, beta adrenergic blocking drugs. Beta blockers enter the bloodstream through the GI tract. They prevent norepinephrine or noradrenaline from binding to the beta adrenergic receptor on the heart cells and walls of blood vessels, causing the heart rate not to increase during fight or flight responses. When you would normally have a fight or flight response, you don't get it. The heart rate doesn't go up. So this is blocks an excitatory response that epinephrine would normally create, norepinephrine would normally create in the heart. As an excitatory response on the blood vessels. Causing them not to relax, not to contract when they would normally contract. So this blocker slows the heart rate by blocking the norepinephrine's effect on speeding up the heart rate. And it therefore lowers, giving the left ventricle more time to fill up with blood. If you're not having to pump your heart so fast, it gives it more time to fill up and then pump, fill up and then pump. When it's beating really fast, it doesn't fill up as much, so it doesn't pump out as much blood. Cardiac output is how much blood is actually being pumped out of the heart with each minute. How much blood is actually being pumped out with each minute out of the heart. And so the higher your heart rate, the higher the heart rate, the less blood being pumped out. So is that right? I should have thought about this. Let me think here. What I want you to remember from this is that it's a beta adrenergic block. So it prevents acetyl norepinephrine from stimulating the heart and causing it to the heart to contract faster. Autonomic tone. This is the normal balance between the rate of these two combating parasympathetic and sympathetic nervous systems. So it's a normal background rate of activity between these two that represents this balance between them to allow for the body's needs. The sympathetic tone keeps most vessels partially constricted because it keeps it partially activated all the time and maintains the blood pressure. Parasympathetic tone maintains the smooth muscle tone in the intestines and holds a resting heart rate, relaxes it. Autonomic tone, so vasoconstriction. There's a strong sympathetic constriction. A little bit, always a little bit. There's always a little bit of an activation, a subtle activation of the sympathetic nervous system that maintains a steady state of partially constricted blood vessels. Smooth muscle contraction resulting in vasoconstriction. A weaker sympathetic activation will result in smooth muscle relaxation and vasodilation. There's a regulated feedback between the spinal cord and the brainstem, regular feedback loops. With input from the limbic system, remember that tends to be your emotional stress responses, and oversight by the hypothalamus. Caffeine, we were talking about nicotine before. Caffeine mimics adenosine. You remember adenosine had to do with ATP, adenosine triphosphate, cyclic adenosine monophosphate. Caffeine mimics adenosine, which is an excitatory neuromodulator. The binding of adenosine to a neuron results in increased acetylcholine by cholinergic neurons, leading to vasoconstriction. That excitatory. Caffeine lowers the neuron's ability to initiate action potentials, decreasing the acetylcholine secretion. And the result is that you have increased vasodilation, meaning increased blood flow. So the reason that caffeine is as addictive as it is because it mimics adenosine as a neuromodulator and lowers the neuron's ability to produce action potentials that is going to effectively lower that neuron's ability to secrete acetylcholine. And when you have less acetylcholine, you have more vasodilation. That's the end of this chapter. Questions? Thoughts? Let's start on to the next chapter. So like I said before, we're going to start chapter 11 and we're going to come back to chapter 16. We have 2 more chapters left. That's chapter 11 and chapter 16. Your exam 3 is going to cover up through chapter 11. Your final exam is going to cover chapter 16 and the rest of it will be comprehensive. I am guessing that before Thanksgiving, which is what, next week, we will finish chapter 11. And so I will schedule exam 3 for over the holiday. I don't expect a lot of you to study all your whole holiday. But I will schedule it so it will be open during that time.
Do we have regular class on Monday and Wednesday next week?
I don't know about Wednesday. I need to look at the school schedule. I don't think we will. And the reason, but I need to look at the school schedule. Bear with me just for a second. Where is the school schedule on here? Do you see it? School year, academic calendar. I know it's on here somewhere. Do you guys see it? Let's see, student. Academic calendar.
Oh, it's on your left.
Aha. Well, that was really not helpful. Oh, there it is. It's down below. So 26th through the 29th. There's that. Bear with me, guys. What does it say on the syllabus? Because I don't think we will, but I don't want to say that for sure until I look at the syllabus.
There isn't that date on the syllabus. November 25th isn't on the Wednesday syllabus.
That is probably because we don't have class that day. This is a little off. We are going to do this lab. We're not going to do this lab on this Wednesday. Wait, what were we off a day? Wednesday, November 4th. We're off a day. I don't know if we do vision at all. You guys just be, I think we're going to go right into this. Obviously, I made a mistake here with the syllabus, but we're going to go right into this. And no, the reason the day isn't there is because, no, we don't have lab next week. We will have class on Monday, but we won't have lab on Wednesday. Does that help?
Yes, thank you.
You're welcome.
When is technically like our final class or like our final due?
The final day is November 30th.
Okay.
So we have 2 more days. So you guys, before I run myself out of time, I'm going to jump into muscle really quickly here. Any other questions? Leave them until after class, because it looks like we're coming, you can tell we're coming down to the end. So the last day of class is actually the Monday after Thanksgiving. So we have the rest of today, we have next Monday, and we have the next Monday. And those are the last days of class. Bear with me, I'll let you know more on Wednesday with regards to scheduling. Yeah, if you didn't think we were coming down to the end, your case study, your 3rd case study is posted. I think it's actually an easier case study, but you may have to Google a little bit to find out some of the answers. But it is posted for you. So, skeletal muscle or muscle tissue. Overview of muscle tissue. Body movement results from alternating contraction and relaxation of muscles. The skeletal system provides leverage and a supportive network for this movement. Myology is the scientific study of muscles. There are 3 types of muscle tissue. There's skeletal muscle, which is found throughout the body attached to the skeleton. It's cardiac muscle that's found only in the heart. There's smooth muscle that is found within the walls of the thoracic and abdominal viscera and surrounding blood vessels. Skeletal muscle is found attached to the skeleton. It functions in motion, example, walking and running, maintaining posture, producing heat allowing you to shiver, thermal regulation. Appearance, it has multiple peripherally located nuclei. Paisley, stop. And it's striated, has an alternating light and dark bands observed under the microscope. Voluntarily controlled, also controlled subconsciously to some extent. For example, you don't have to think to relax and contract your diaphragm to allow you to breathe. You don't have to think to contract the muscles that allow you to maintain your posture and keep your head up. Cardiac muscle is located within the walls of the heart, has the function of pumping blood, has one centrally located nuclei. It is striated, contains intercalated discs that are gap junctions. Gap junctions are little tunnels or little pathways that connect the cells together to allow them to be joined together and have direct communication between the cells. Control, it's involuntarily controlled, displays autorhythmicity through that sinoatrial node. It can speed up or slow down when influenced by neurotransmitters and hormones. Smooth muscle, located within the walls of blood vessels and hollow internal organs and structures. Example, it's in the GI tract. Skin, it's attached to hair follicles. Function, it is involved in peristalsis, which is the rhythmic contracting and relaxing. Rhythmic contracting and relaxing of the GI tube that allows substances to move along the tract. Regulates organ volume, temporary storage of food and urine due to control of bands of smooth muscle called sphincters. Parents, it's one centrally located nuclei, just like that in the cardiac muscle. It's smooth, it doesn't have any striations. It's involuntarily controlled, regulated by the autonomic nervous system, neurons, and hormones.
Hey, Professor.
Yeah.
We learned about these in another chapter, right? But it was just more in depth. This is kind of like an overall, like where they are and what they do, right? Lab, right.
Yeah, so a lot, this is kind of a coming back to it.
Okay, I just wanted to make sure. I'm like, hey, I actually remember this.
Good. Well,
I was gonna say the same thing.
That's good, guys. That's good. If it's starting to come back to you, that's not a bad thing. That's good.
Yeah, I was like, I definitely remember doing some of this before.
Good. So this is just a compare and contrast of the different muscles. I haven't put a review worksheet together. I haven't done it for the autonomic or I felt like it was probably more important to maybe grade your case studies. But I will get on them as soon as I can. Skeletal muscle fiber development. Each skeletal muscle fiber arises during embryonic development from the fusion of 100 or more myoblasts. So if you remember that the skeletal muscle contains multiple peripherally located nuclei. The reason is because multiple up to 100 individual cells are fusing together to form this type of muscle fiber, meaning that there are going to be 100 or more nuclei. Each mature skeletal muscle fiber has over 100 nuclei. Satellite cells are unspecialized myoblasts that retain the ability to regenerate when muscle fibers get damaged. Cell division ceases once fusion occurs. The number of skeletal muscle fibers is set when you are born. Once this fusion occurs, there's no more cell division of the muscle fibers, muscle cells. So most of these cells will last through the lifetime of the individual. Muscle growth occurs by hypertrophy. So the muscle enlarges. There's an enlargement of the muscle fiber that exists, but you don't develop more muscle fibers. They only get bigger. Testosterone and human growth hormone can stimulate hypertrophy. Some dominant properties of muscular tissue. It's excitable. Has chemical signals that stretch and electrical changes across the plasma membrane. Conductivity, local electrical change that triggers a wave of excitation that travels along the muscle fiber. They can generate and propagate muscle action potentials. So two properties, excitable, responsiveness, conductivity, contractility, ability to shorten when stimulated. There are two different types of muscle contractions. The muscle develops tension but does not shorten. That's an isometric. Iso meaning the same, metric meaning length. So isometric meaning the same length. It stays the same length contraction. So tension develops, but it doesn't shorten in length. Isotonic muscle contraction is when there's enough tension is generated in the muscle to move an object, so the muscle shortens. So the tension is the same. Isometric, the length is the same. . it's some other properties of muscular, okay, so 2 properties: excitability, conductivity, contractility. Isometric, meaning the muscle develops tension, but it does not shorten. It develops enough tension that the muscle shortens. Extensibility, capable of being stretched between contractions without being damaged. Elasticity returns to its original resting length after being stretched. We're going to talk about this one when we talk about muscular dystrophy. This is where the one property that their muscles struggle with is extensibility. Capable of being stretched between contractions without getting damaged, their muscles get damaged. Elasticity returns to its original resting length after being stretched. So those are the 5 properties. Which of the following is not a function of skeletal muscle tissue? Moving your eyes from left to right as you read this question, holding your head up, shivering to generate body heat when you become chilled, moving food through the small intestine, stabilizing joints and helping to maintain body positions.
Maybe D.
Because D is predominantly smooth muscle. D is smooth muscle. Muscle is organized with a fascia that is a sheet or band of fibrous connective tissue that is deep to the skin, but it surrounds kind of like it's a sheet. So it's kind of like, what do you call it? What's the gauze tape that, an ace bandage? Kind of like an ace bandage that surrounds the muscles and other organs of the body. Kind of holding them together and keeping them organized. Just a sec, buddy, I'll take you out in a minute. The subcutaneous layer stores fats, separates muscle from skin, and protects muscle from trauma. This fascia lines the body wall and limbs, holds muscles with similar functions together. You guys let me go grab him because he's needing to go out, but my roommate isn't here. So let me just go grab him for 1 second here. Hey. It's amazing how much a 3-pound dog can make noise. He gets going and it's amazing how much noise he can make. So fascia lines body walls and limbs, holds muscles with similar functions together, kind of like an ace bandage, allows free movement of muscles, and carries the nerves and blood vessels and lymph vessels. It's lining. Fills the spaces between muscles. It is what these fascioles are what give meat, M-E-A-T, its characteristic grain. Around the skeletal muscle fiber cell, remember muscle fibers are cells, muscle cells. The sarcolemma is the plasma membrane of a muscle fiber. Right here. The sarcoplasma, remember sarco is muscle. The sarcoplasm is the cytoplasm of the muscle fiber. Myofibrils are long protein cords occupying most of the sarcoplasm. Glycogen is the carbohydrate for ATP synthesis. Glycogen is branched glucose molecules that can be, and the glucose can be broken down and used to make ATP. Myoglobin is a red pigment that binds and releases oxygen for ATP production. So 3 major components in the sarcoplasm: myofibrils, glycogen, and myoglobin. Mitochondria lies in rows and is located close to the contractile muscle proteins that use ATP during a contraction, so ATP can be produced as quickly as needed. The smooth, let's see here, the sarcoplasmic reticulum is a modified smooth ER that forms a network around each myofibril. It acts as a calcium reservoir and releases calcium through channels to activate contraction of the muscle. It has terminal cisternae that are dilated end sacs which cross the muscle fiber from one side to the other. Transverse tubules are tubular infoldings of the sarcolemma. Remember, that's the plasma membrane of the muscle cell. Tubular infoldings, so indentations into the cell, that are tunnels formed through which action potentials can travel and spread throughout a muscle fiber. So a triad is the transverse tubules and the terminal cisternae of the sarcoplasmic plasma reticulum. Any questions? Stay with me for just a few more minutes here. Myofibrils are the contractile organelles of the skeletal muscle. They contain both thin and thick myofilaments that are arranged in units called sarcomeres. Striations make the entire skeletal muscle fiber appear striped. So sarcoplasmic reticulum, remember the terminal cisternae of the sarcoplasmic reticulum, the transverse tubules together make up a triad. Sarcomeres are compartments of arranged filaments which extend from one Z disc to the next Z disc. This is a Z disc, this is a Z disc. So the sarcomere is this compartment from one side to the next. Which structure releases calcium to trigger the muscle contraction? Do you remember?
It's calcium,
Yeah, well, which structure releases the calcium?
bone,
No. Well, what about right here in the muscle cell? What about the sarcoplasmic reticulum? Acts as a reservoir and releases calcium through channels to activate contraction. Why does calcium have to be actively transported back into the sarcoplasmic reticulum? Because we also want the muscle to be able to relax. And if it's not able to get rid of the calcium, it's trapped within this space. If it's not able to get rid of it, the muscle is going to consistently contract and not be able to relax. So 3 types of muscle proteins comprise the myofibrils: contractile proteins that cause the muscle shortening when they interact with one another, which are myosin and actin; regulatory proteins, which regulate the interactions between contractile proteins, which are tropomyosin and troponin; structural proteins, which keep the filaments in proper alignment and give myofibril elasticity, links the microfibrils to the sarcolemma. Titin, alpha actin, myomesin, nebulin, and dystrophin are these structural proteins. Guys, I think I'm going to stop right here. Any questions, thoughts?
No, it's basically just a recap of what we were already learning. It just seems a little more in depth. So
Yeah, I'm glad you remember as much as you do, because that will make this easier.
yeah, definitely. I'm like, why couldn't all of them be like this? You learn one and then just reevaluating it.
It's gonna get more complicated, but this is a good. I'm glad that the beginning is a review and it's not like hitting you out of nowhere. All right, I will see you guys on Wednesday.
All right, sounds good, Professor. Thank you. Have a good day.
General, tell them to have a good day, okay? just puts up with it.
I just wanted to let you know, Professor, that the case study 1 that I submitted that you marked as a 0 because I submitted the
Yeah,
chapter.
Kyle. Yeah,
I just submitted the correct.
I got it.
My labels. I work from a Mac, and the labels of my documents and stuff are really weird, and so I can't always. Sometimes it just pops up as like biology, and I can't see what assignment it is. So
Kyle, you have a lot of missing assignments.
yeah.
What can I do?
I'm working on turning in quite a few of them this week.
Why don't you just stay in communication with me? If you can't get one of them to allow you to submit it, let me know. Okay?
Okay. Have a good day.
You too.
Professor,
Yeah.
can you check like what assignment I've been missing?
Yeah, give me 1 second.
And I submitted my work state 8, 9, and 10 last week.
Okay. I've just been grading case studies. I haven't graded anything else, but I will get to it. Let me lock my share here. Stop sharing. Bear with me just for a second here. Okay. So, the Smart Book assignment for chapter 15. The Smart Book assignment for chapter 14. The connect activity for the nervous system, the monosynaptic reflexes.
It was due like probably last week, right? Can I still do it?
Well, I think I extended out the deadline for it, so I think you should be able to do it. Let me, I'll go in and look. Give me 1 second here. The lab quest 4 and the second case study.
Did you say lab quest 3 or
Lab
4?
quest 4.
Okay.
Let's see here, what else? The chapter 15 quiz, but I just put that out. And the case study 3, which isn't due yet, but it's out. Let me come in and look at them. Going to McGraw Hill. Yeah, it's not due until the 23rd, that nervous system connect activity. So
Okay.
it's still open.
Okay, thank you, Professor.
You're welcome. Have a wonderful day, okay?
Thank you, you too.
Hi, Professor.
Hi.
Can open chapter 14 for reading and connect. We didn't close yesterday. I didn't finish it actually.
Let me, yeah, I can do that. Give me 1 second here. I need to remember which which class here. This one is for
Thank you.
walking, of course. This is this one. Okay. 14. I'm gonna set it up for another week, okay?
Yeah, that's sure. Thank you very much.
So it'll be through the 22nd,
Oh, okay, thank you.
because I think that was also one that Ditmo needed to do too. So they're both set up for that.
Okay.
Okay. Have a wonderful day, all right?
Thank you, same to you.
Thank you. Kelsey, are you there? Sabrina? Kelsey or Sabrina, are you there?